Hi everyone, this is the fourth lecture for your chapter Cell. In the last class, we discussed prokaryotic cell. We discussed all the different types, how the structure is, what are its components. We also discussed the different types. Okay, we saw cyanobacteria, archaebacteria, and then eubacteria, or what you normally call as bacteria. Fine. Today we will be starting with cell wall. Cell wall is outer rigid protective supportive and semi-transparent covering. Okay, it is outer, it is it is the outermost uh, layer of a plant cell or a bacterial cell or a fungal cell. It is rigid, okay, and protective. Its function is to protect the cell. Supportive, it supports the cell, it maintains the cell shape, okay, and it is semi-transparent covering of plant cell. If it were not semi-transparent, then you would not be able to see these cells under the microscope, right? If it were opaque, if the cell wall was opaque, then you would not be able to see such kind of cell structures under a microscope, right? Because cell wall is present in 3D. It is a covering over the cell, and cell are not 2D, okay? They are 3D structures. Fine. They are found in fungus, prokaryotes, especially bacteria, which we have discussed. Some protists are unicellular, eukaryotes, okay, it includes yeast and all. So in yeast also, which are small unicellular fungal cells, but they are considered in protista, so they also show cell wall. Fine. The composition is different, okay, though all of them are called cell wall. Plant cell wall, fungal cell wall, prokaryotic cell wall, and protist cell wall are different. Fine. Cell wall was first seen in cork by Hooke. Okay, we have already studied this, Robert Hooke. It has thickness which varies. So its thickness, it varies between different types of cells from 0.1 micron to 10 micron. This thickness can be different in different kinds of cells. Cell wall is a non-living extracellular secretion of a cell. So it is non-living, okay, it is just a structure, at least it acts as a structural barrier. Okay, it has no role as such, direct roles are not seen, but its function is to be a structural component, like a brick wall in your house. Okay, if you see in your house, there are brick walls which do the function of protecting you from the environment, right, or compartmentalization. So cell wall, it is non-living. It is extracellular secretion of the cell. So the cell itself is secreting the cell wall. Okay, it is secretion of a cell. However, it is capable, or it is metabolically active and capable of growth. You can see there is increase in cell wall deposition. Okay, the cell wall can become thinner or it can become thicker. Then it can grow, especially if you remember auxin. So in effects of auxin, you have studied cell elongation. How will the cell elongate if the cell membrane is rigid? Sorry, cell wall is rigid. So the cell wall itself can also grow. Okay, because it grows, the cell can grow a little bit, not as much as animal cells grow, but plant cells or their cell walls can also grow. Okay, they are also metabolically active in the sense that some reactions can take place in the cell wall also. Cell wall of plants consist of cellulose, hemicellulose, pectin, and protein. Here, cellulose, hemicellulose, and pectin, all three of these are polysaccharide. Polysaccharide means they are long chains of sugar molecules. Okay, it could be glucose, it could be fructose, glucose and galactose, etc. For example, if you remember, cellulose is a linear polymer, or it is a polysaccharide of beta glucose. Okay, so beta glucose will come together, they will form a long chain, and that will form the cellulose. Same way, there is hemicellulose, pectin, all three of them are polysaccharides. And finally, you have proteins also which are present in the cell wall. Besides cellulose, the algal cell wall. Now there is some difference between the plant cell wall and the algal cell wall. Okay, so this is an important point. Remember it. Algal cell wall has galactans. Galactans are polysaccharide made up of galactose. Mannans. Mannans are polysaccharide made up of mannose. Okay, mannose is also a sugar, 6-carbon sugar, so hexose sugar. Galactose is also a hexose sugar. Glucose, galactose, and mannose are epimers. If you have studied, well and good, so they are basically isomers. If you have not studied, when we will cover biomolecules, we will study, do not worry. Okay, so algal cell wall has cellulose. Okay, so it is written beside cellulose, so cellulose is there. Apart from cellulose, there are galactans, which are galactose polysaccharides, mannans, which are mannose polysaccharides, and calcium carbonate, CaCO3. Okay, this all of these will together form this tough outer cell wall. Why it is different? Because algae are normally growing in water conditions. Okay, they normally grow in either pond or where there is flow of water. Fine. So their cell wall are supposed to be a little more tough compared to the plant cell wall. Okay, normal plant cell wall. Fine. Now the cell wall is made up of 3 parts. It is made up of matrix. Matrix is ground substance, okay, something that is filler, like how cytoplasm is just filling up the space between different organelles. Same way matrix fills up the space between different parts of a cell wall. Okay, here you can see a cell wall is made up of many fibers and small and large things, but if you see, there is a lot of space also in between. So this space is not empty, it is not filled with air, it is filled with matrix. So matrix is 60% water, so it is majorly made up of water. It has 5 to 10% hemicellulose. Okay, these green colored fibers you see are hemicellulose. They are part of matrix. 2 to 8% pectin. Okay, these yellow colored fibers that you see are actually pectin. 0.5 to 3% lipids, very small amount of lipid is also present, and very small amount of proteins, which you see here. Some soluble proteins are present in the cell wall, okay, and present in the matrix part of the cell wall. Then come the microfibrils, micro meaning small, okay, and fibrils because they are fibers, but smaller fibers are called fibrils. Clear? So microfibrils, they are normally made up of cellulose. So these blue colored cylinders that you see are actually your cellulose microfibril. Okay, so 10 to 15% of them are made up of either cellulose or fungal cellulose. 10 to 15% meaning the major portion of cell wall is matrix, and after that 10 to 15% is made by cellulose microfibrils, or in case of fungus they are made up of chitin. Chitin is also called fungal cellulose. Chitin is nothing but it is again a polysaccharide, okay, homopolysaccharide. And then there are some other ingredients apart from matrix and the microfibrils. There are some other things also like lignin, cutin, suberin, silica. Silica is SiO2, sand, minerals like iron, calcium carbonate, etc. As we saw in the case of alga, there is calcium carbonate. Some have waxes. Okay, waxes are nothing but they are long length or long chain hydrocarbon with a small alcohol group. Okay, some of them have alcohol group. So waxes, which are hydrocarbons. Then tannin, resin, gum, these are all secondary metabolites. If you know what primary and secondary metabolites are, very good. If you do not know, we will be covering them in biomolecules. Okay, so do not worry. But I'll just give you an example. Have you ever tasted or seen green tea? And if you put that green tea in water, it releases some green or brown colored compound, right? So those are mostly tannins or resins. Resin, I'll tell you again. Have you seen ember? Or from some trees, you will see some gum like golden colored fluid flowing through. So that is resin. And in case of some, there is gum. Then some have that milky secretion. Also, there are rubber plant which secrete latex, right? So some such resin, tannin, gum are also present in this cell wall. Okay, so this is the basic composition of a cell wall. What is a cell wall made up of? It is made up of matrix, which is mostly water, hemicellulose, pectin, lipids, and proteins. It is made up of microfibrils, which are either of cellulose kind or fungal cellulose kind. In case of fungus, they are fungal cellulose. In case of plant, they are cellulose. And there are some other ingredients also present depending on the type of plant, okay, like lignin, cutin, suberin, silica, etc., etc. We will see them, the important ones at least, in this lecture. Now we move on to the structure of cell wall. We saw the composition of cell wall. We will be discussing the structure of cell wall. It can have up to 3 parts. Okay, a cell wall has 3 parts. It has middle lamella, primary wall, and secondary wall. Fine. Here the black portion you see is nothing but middle lamella. Then near the black portion, what you see is the primary cell wall. It is very thin. And after primary cell wall or primary wall, there is this thick layer. It is called secondary wall. Can you see here? And after the secondary wall, the innermost layer is of plasma membrane. Fine. So it is made up of a minimum of 3 parts. It has middle lamella, it has primary cell wall, and secondary cell wall. Not all plants will have secondary cell wall. Okay, so they may have middle lamella and primary cell wall. We will see. So middle lamella is thin. You can see it is very thin. It is amorphous and cementing layer between 2 adjacent cells. It is like a cement, okay, between 2 bricks. When you put cement, same way you put middle lamella between 2 adjacent cells. Adjacent matlab baju wada cells. Fine? Neighboring cells. It is the first layer which is deposited at time of cytokinesis. Just a quick recap. During cell division, the plant cell divides by forming a cell plate. Okay, it forms a cell plate. The cell plate is formed by something called phragmoplast. This phragmoplast will come, it will start depositing the cell plate. Cell plate is nothing but it is made up of calcium and magnesium pectate. Okay, pectin, you know, it is a polysaccharide. So this polysaccharide, when it is in relation with calcium or magnesium, it forms calcium pectate or magnesium pectate. Okay, salt, sort of a compound rather. So that is what forms the cell plate. This cell plate will keep on growing, and this is how the 2 cells divide. So this cell plate is the one which is middle lamella. So middle lamella is the first layer deposited at time of cytokinesis. Cytokinesis is division of the cell. Clear? It is made up of calcium and magnesium pectate. Right, now one more thing. The softening of ripe fruits is caused by partial solubilization of pectic compounds to produce jelly-like consistency. Think of a raw mango, okay? A raw mango. How hard is it? It is very hard, right? You have to literally chew on it. Then it is very sour. It has no flavor as such of mango, right? It is just khata. Now, as the fruit ripens, so softening of ripe fruit happens. That same hard mango fruit can turn into a very sweet and very soft yellow or orange colored mango, right? This happens because the middle lamella or pectin between the 2 cells, it is solubilized, okay? It is dissolved by pectinase and other enzymes. So these enzymes, they slowly degrade such pectin, and when the pectin is dissolved, the fruit or the so the fruit and the cells, they become soft. Okay, they become less rigid, they become soft, and it produces jelly-like consistency. I'll tell you one more thing. you know, the juices like apple juice. Apple juice, if you try to make at home in mixer grinder or something, then it will be very cloudy. It has a lot of particles. Or if you see guava juice, the guava juice also has a lot of particles. It is opaque. So these particles are mostly because of pectin. Okay, pectin and other polysaccharides also, fibers also, but pectin is the important one. So what they do is they introduce pectinase enzyme, and this pectinase enzyme will degrade or solubilize this pectin, and then you get clear apple juice. So what you get in Tetra Pak is very clear. It does not have those particles, right? So because it has been treated with pectinase. In jelly and jam industry, they use this pectin, okay, to make the jam and the jellies thick. So it is used like that. So that is the role of pectin. And here we have discussed the plant, middle lamella. Fine. I hope you have understood. Middle lamella is the first layer formed between the two cells. It is made up of calcium and magnesium pectate. Then comes the primary wall. Okay. So around this magnesium pectate or calcium pectate middle lamella, the next layer what you see is again secreted by the cell itself. Okay. Cell 1 will secrete its own primary cell wall. Cell 2 will secrete its own primary cell wall. Okay. So it is the first formed wall of a cell produced inner to the middle lamella. Okay. It is produced inner, on the inner side of the middle lamella. It consists of cellulose or fungal cellulose microfibers embedded in matrix. This part we have already discussed, but let us understand this diagram once again. You have the outermost layer of a cell to be middle lamella. Okay. Here also you can see the outermost layer is middle lamella, then it is followed by primary cell wall, followed by secondary cell wall, and then plasma membrane, and then you have the cytoplasm. Okay, so you can see here the outermost layer is middle lamella. Middle lamella is made up of pectin, right? Pectates, calcium and magnesium pectate. So it is basically made up of pectin. These yellow fibers that you see, it is very thin. After that comes the primary cell wall, which is secreted by the cell itself. It is made up of cellulose microfibrils and it has matrix. In matrix, there will be other hemicellulose, pectin, protein and lipid molecules also. Now here, look below. This is the diagram of a fungal cell wall. Okay, in case of fungus, there is fungal cellulose or chitin which is present. The green colored portion you see here is made up of chitin. And this here is the cell membrane. So above the cell membrane, there is chitin present. Fine. It grows by intussusception or addition. Okay, intussusception, sorry, intussusception or addition. So how does this primary cell wall grow? So it is by addition. Okay, the cell will secrete cellulose. There is an enzyme called cellulose synthase. Okay, and the cellulose synthase will keep on secreting cellulose. Okay, and that is how these cellulose fibers are made. And they keep on adding the cellulose fibers. Okay, so by addition of material to the existing wall, intussusception. Clear? Intussusception. Remember this word. It is a little difficult, but it will be fun. If you pronounce it 3-5 times, you will remember it. The size is very thin. Primary cell wall is very thin. Just remember this diagram. Okay. This is middle lamella. On the middle lamella, this small layer you see is the primary cell wall. It is very thin. It is about 0.1 to 3 micron in size. But it is capable of expansion. How will it expand? It will expand if you keep on adding the material to the existing wall. Some cells possess only primary wall. Okay, some cells have only primary wall, no secondary wall or nothing doing. They have middle lamella and they have primary cell wall. Such cells are cells of leaf, fruit, then cells of cortex and pith. Basically, parenchyma cells, parenchymatous cells are the ones which have only primary cell wall because they do not require more rigidity or structural support. a small primary cell wall is enough. Okay. Now secondary cell wall. Secondary cell wall is produced in some mature cells when they have stopped growth. Okay, growth. When they have stopped growing, so cells, they stop growing. After that, there is deposition of secondary cell wall. Examples of such cells are tracheids, which are parts of xylem, vessel elements, which are again parts of xylem, fibers. Just think of fibers like jute fiber or some other fibers that you know. Okay, so such kind of fibers. Then collenchyma. All of these will have secondary cell wall. Okay, it is by accretion or deposition of material over the surface of existing structure. Okay, the size is 3 to 10 micron, so it is thicker than the primary cell wall, and it is made up of 3 or more layers. Okay, secondary cell wall can be in layers. Okay, one by one by one. So such layers are named S1, S2, S3, etc., 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 depending on the number of layers. Mostly plants have 3 layers, but some rare exceptions have more than 3 layers of secondary cell wall also. Right, now let us see in this diagram, let us compare. This here, what you see is your normal cell wall, where you have middle lamella shown in brown. Under middle lamella is your primary cell wall. Okay, primary cell wall, the same thing, pectin, hemicellulose, cellulose, proteins, and some lipids. Fine, that is how the cell wall is made. Here the microfibrils of cellulose are arranged haphazardly. They are arranged in any manner they want. Okay. Now look at the bottom diagram. Here the uppermost brown block is of middle lamella, and beneath it the orange layer is the primary cell wall. So can you imagine, this whole structure is so small, it is so tiny compared to the secondary cell wall. The secondary cell wall is really thick. It is made up of cellulose microfibrils, but they are arranged very particularly in a very uniform manner. They are arranged in a uniform manner. And beneath this secondary cell wall will be the plasma membrane and then the cytoplasm. The innermost layer of secondary wall has presence of xylans. Xylans are again some chemical compounds. And if they are present, then it is called tertiary cell wall. Very rarely you see it, but they are called tertiary cell wall. Example is tension wood in gymnosperms. Okay, in gymnosperms there is tension wood which has xylans or which has tertiary cell wall. The composition of cell wall is basically similar to primary cell wall. Okay, secondary cell wall, primary cell wall, their components are the same. You have matrix, you have hemicellulose, cellulose, pectin, etc., etc. Same things. Fine. Yeah. Okay. One difference. Sorry. The pectin will be present in the primary cell wall, but it will not be present in the secondary cell wall. Okay. So that is the difference. Instead, some other molecules will be present. We will see what all is present. Fine. A number of different materials may be deposited in the wall. Okay, there may be different materials deposited, like lignin. Lignin, here you can see this structure. Okay, the gray colored boxes. So this lignin reduces water content of the cell wall matrix. Now matrix is 60% water, but lignin will reduce the 60% and increase the hardness. It will increase the toughness of the cell wall. Okay, but it will not affect the cell wall permeability to water. It will still allow water to pass through. Okay, it will allow the water to come in the cell and go out of the cell. So though it is reducing water content of the matrix and it is toughening the cell wall, it is still allowing water to pass through. Clear? Then you have suberin. Suberin is another type of fatty substance. It is found in cork. Cork, the same cork that we saw in Robert Hooke's description, right? So when we will study anatomy portion of plants, or you might have studied, then you know what cork cambium is and what cork is, right? So cork and endodermal cells of a root stem, okay, root and stem, they have this substance called suberin present in their cell wall. The suberin makes it impermeable to water. Suberin will not allow water to pass through. Okay, this layer is your endodermis. Okay, epidermis, then you have cortex, then you have endodermis or pericycle, then you have stele. Anyway, right. So, yeah, we were talking about suberin. So this is the cell wall. You have middle lamella. ML means middle lamella, outermost layer. Then you have primary cell wall or primary wall, PW. Then you have secondary wall. The secondary wall has a lot of suberin. Okay, and the suberin will not allow water to come in. Then you have plasma membrane and cytoplasm. Fine. So suberin is responsible for making the water take symplast pathway instead of apoplast. You will also study that in transport chapter. Okay. Then you have the third deposition, which could be of cutin. Cutin is again some sort of fatty substance which is present in epidermal cells, okay, the outside cells. That too in leaves, you can see them outside as well. Okay, cell wall ma deposition hase, પણ બહારની તરફ પણ deposition hase. So you will see the deposition of cell wall, cutin in cell wall as well as outside the cell wall. Fine. So if it is outside the cell wall, it is called cuticle. Okay, this cuticle will create a layer and it will reduce the transpiration. Transpiration is loss of water in form of water vapor. So it will avoid the loss of water by water in form of water vapor, or what is called transpiration. Clear? Okay. These are the layers. The outermost layer of a cell is middle lamella. Then comes the primary cell wall or primary wall. Then you have secondary cell wall, which could be S1, S2, S3, etc. Okay, outermost will be S3, S2, S1. Then you have cell membrane, and innermost will be your cytoplasm. Clear? These are the general plant cell wall layers. Now between the cell walls, there is connection. Okay. Now if there is, let us say, suberin and all, which will not allow water to pass through from one cell to another, then how will water get absorbed, or how will water reach each and every cell? Right? So there should be some connection. So these connections are called plasmodesmata, or singular is plasmodesma. Plasmodesma are cytoplasmic connections. Okay. Here what you see, these are the cell walls, and between the cell walls there is a hole. Okay, there is a hole, and this hole, through this hole there will be a connection between the two cells. This is cytosol of cell 1 and this is the cytosol of cell 2. Here also this is the microscope, electron microscopic diagram, and this is the cell wall. You can see in between the cell wall there are some connections, okay, between the two cells. This is cell 1, this is cell 2. They form a protoplasmic continuum called symplast. Okay, they form a continuous or continuum, protoplasmic, meaning protoplasm of one cell is connected to the other, and this is called symplast. A plasmodesmata is 40 to 50 nanometer in diameter. It is very small. Okay, it is 40 to 50 nanometer in diameter. It may be simple. Okay, here what you see is very simple, or branched reticulum. There should be a space in between, my fault. Okay, branched meaning instead of this one there will be multiple branches. Now let us come to this point. What is this one thing you can see in the pore? So this is actually smooth endoplasmic reticulum. Smooth endoplasmic reticulum of adjacent cells are connected. Okay, and they are connected by this small tube-like structure which is called desmotubule. Okay, desmotubule is a connection of smooth endoplasmic reticulum of one cell with another cell. Clear? It could be this single tube or there could be multiple tubes. Fine. So that is what is meant by branched reticulum, endoplasmic reticulum. Okay. Or it could be just a simple one like this. Plasmodesmata form channels for controlled passage of small sized particles. If the particle size is less than 40 or 50 nanometer, it can pass through from one cell to another. This is how the cells communicate with each other. As well as there is transfer of some specific signals. If there is a signal, then it will be transferred either using this pore or using this desmotubule, okay, from one endoplasmic reticulum to the other. From one cell to the other, the signal will pass through, the message will be passed through. Here you can see this is a simple plasmodesmata, and these are the complex plasmodesmatas. Okay, you can see they form either a Y shape or a K shape or a double I type of shape. Okay, so these are all complex, but this one is a simple one which we saw here. This is a simple plasmodesmata. The brown colored portion is cell wall. The blue colored portions are the cytosol of cell 1 and cell 2. And this portion is endoplasmic reticulum, okay, the desmotubule. Here beneath you can see a complicated diagram also, that when the cell divides, there is a plasmodesmata between the two cells. As they mature, they form, there is a simple plasmodesmata, okay, and they form this cross. Cross means complex plasmodesmata later on in the life. Then later on, as the cells keep on dividing, they form these complex connections between them. Okay, if there are 4 cells, they might be connected like this. Okay, so these are the differences between the two. I want you to write it down. Okay, one way to be more productive, okay, or to learn more from less effort is to make a comparative table. This is my top tip for you. If you make a table like this, there will be a lot of benefit. What benefit? First thing, because the sentences will be small, you can refer to them quickly. Okay, easily you will be able to read this one slide and compare whatever we studied during the whole lecture. Fine. So from these small segment, you will be able to learn a lot. So always make a habit of making such type of tables. So you have a table. Then in here you will study individually, as well as you can compare primary as well as secondary cell wall. What is the difference? So studying it individually and studying it while comparing is also another way of remembering it better. Okay, so this type of tables will be very helpful for you. So if you are making notes for NEET, I suggest you start making such type of tables, so it will become easier for you to refer. Okay, textbooks get a little boring after a while. How much of it will you read? Even litho or whatever material they are giving you, how much of that will you read? You get bored. But if you have it in such point wise manner, very nicely written, you will be able to remember it well, right? So let us discuss very quickly what is the difference between primary cell wall and secondary cell wall. Primary wall is laid inner to middle lamella, while the secondary wall is laid inner to the primary cell wall. Fine. It is primary cell wall is formed in young growing cell, while secondary cell wall is formed after the cell has stopped growing. Primary cell wall is capable of extension. It grows by intussusception or addition of materials. Okay, and here the secondary cell wall is not extensible. Not extensible matlab, if you add auxin, then you will not see cell elongation. Okay, there will be no elongation. It is not extensible. Like how a rubber band can extend and come back, that will happen in primary cell wall. But the secondary cell wall is very thick. It is very organized. It will not extend. It is not extensible. It grows by accretion or deposition of material on existing structure. Primary cell wall is single layered. Secondary cell wall has 3 or more layers. Hydration is 60%, while the secondary cell wall has less hydration. It is 30 to 40% only. Clear? Cellulose content is comparatively low in primary cell wall. It is 10 to 15%, while the cellulose content is comparatively high in secondary cell wall. Okay, secondary cell wall, because it is thick, it has a lot of cellulose also. Cellulose fibers are short, wavy, and loosely arranged. They are very haphazardly arranged. Okay, they are small in size, shorter in size, they are wavy, and they are loosely arranged. While in secondary cell wall, they are long, they are very closely, very tightly arranged in straight and parallel manner in secondary cell wall. Hemicellulose content is high, it is up to 50% in primary cell wall, while hemicellulose content in secondary cell wall is 25%, less. Lipid content is 5 to 10%. In the secondary cell wall, lipids are almost absent or negligible. Additional chemicals present are lignin in primary cell wall, while in secondary cell wall there can be lignin, suberin, cutin, pectin, a lot of things. Primary cell wall is very thin, it is 0.1 to 3 micron in size, while the secondary cell wall is 3 to 19 micron per micron in size. Okay, these numbers obviously differ from book to book, but it is just given for you to understand. Okay, it is just for logic, for reasoning, that you come to know secondary cell wall is very thick. Okay, it is larger in size compared to the primary cell wall. Functions of the cell wall. It protects the protoplasm against mechanical injury. Okay, if you slap a tree, nothing will happen to it. Why? Because it has cell wall. Okay, so that is mechanical injury. It will protect the protoplasm or the cytoplasm against it. It protects the cell from pathogens. Pathogens are disease-causing organisms. Okay, so it protects from pathogens. It provides rigidity and shape to the cell. Okay, one more thing. How will you be able to see a cell wall? Let us say you are looking under a microscope. How do you know it is cell wall and not cell membrane? Okay, first thing, cell membrane is very thin. You hardly ever see it. But when you plasmolyze the cell, that is, you put it in hypertonic solution, water will move out and the cell membrane will shrink while the cell wall remains the same. So here, whatever structure you see will be the cell wall, which remains the same. And whatever shrinks will be your cell membrane and the cytoplasm. So this is how you can clearly tell that that is the cell wall and this here would be the cell membrane. Fine. It provides rigidity and shape to the cell. Okay. It counteracts osmotic pressure. That is, if you put a cell with a cell wall in water, pure water, which is hypotonic, meaning it has low concentration, then water will start entering the cell. But because it has a cell wall, this cell wall will create a wall pressure and will not allow more and more water to come in. So it prevents the cell, plant cells from bursting, okay, or excessive endoosmosis. It gives strength to the land plants to withstand gravitational forces. Okay, against the gravity, when the land plants have to grow, then the cells have to have some rigid or stable structures. So these stable structures are nothing but cell walls. Because plants, as you know, do not have skeleton like we do. They do not have bones. What do they have? They have cell wall. By its growth, the cell wall helps in cell expansion. Okay, the primary cell wall can grow, only it can extend. So it helps in expansion. Some enzymatic activity is seen in cell wall, not a lot, but some. Okay, especially in case of prokaryotes, you can see some enzymatic activity as well as some metabolism happening near the cell wall. Cutin and suberin are present in cell wall. They reduce the water loss through transpiration. Suberin is water. Suberin and cutin, both of them are water impermeable. They will not allow water to pass through, right? So they reduce the water loss. Walls of sieve tubes, tracheids, and vessels are specialized for long distance transport. Now again, have you ever tried drinking some fruit juice or smoothie with a straw? What happens when there is very less amount of smoothie or juice present in the bottom? The straw will collapse. Why does the same not happen with xylem or phloem? They are also straw, right? It does not happen because they have cell wall, and this cell wall is normally lignified. Okay, there is lignin present, which will provide a structure, okay, structural support, so that the straw does not collapse. straw पिचकी ना जाए ना माटे. Fine. Here you can see this is a microscopic diagram of xylem and the protoxylem. Okay, protoxylem is formed during secondary growth. Here is the diagrammatic representation. Okay, whatever these red structures you are seeing, red lines you are seeing, they are actually lignin. They could be either arranged in this spiral fashion or they could be arranged in this circular fashion. Okay, annular lignin. Annulus means ring. Okay, so they can either be in form of ring or they can be in form of such spirals. They provide support to the xylem. Okay, and this will allow them for long distance transport. Okay, some seeds store food in form of hemicellulose in the cell wall. Okay, only in some seeds there will be storage of food in form of hemicellulose, like how we store starch. Clear? Here we end today's lecture on cell wall. Fine. Solve questions. If you have doubt, let me know.